Today, let's look at life science and how is it different from the other sciences. While chemistry will look at matter and the elements that make up matter, all matter, living or non-living, life science concentrates on living matter and all other matter that helps to sustain life. So while you are dealing with living matter, of course, you also look at non-living matter, or generally other matter that are found inside living organisms. So with this, we begin to see new terminologies, new language or terminologies. We begin to see language like, instead of atom, we begin to see what? Cells. Cells will make up what? Tissues. Tissues will make up what? Organs. And then a combination of organs will give us the organism. So from here, we are clear with the science and we are clear with what we'll be looking at. And this is a systematic organization of living beings or living things, from cells to tissue. So we begin to see that the smallest living unit is called what? The cell. So we have some organisms that will just be made up of one cell. So we'll call them what? Unicellular organism. And then those that are not unicellular organism will be what? Multicellular organism. So we begin to see terminologies coming in. Uni, multi. Unicellular. Why is the iPad still on? See the way. Why don't you have your pen? Look at your friends. Right. Now, what other terminology shall we see today? Will be seen as we move forward. First, let's read.
There is nothing more complex.
Read faster.
There is nothing more complex in the entire universe than living things. Life comes in an extraordinarily diverse range of forms, from microscopic bacteria to giant plants and animals. Each organism has specialized ways of living, working, and interacting with each other.
And of interacting.
With its environment.
Now, here we have seen some items that we have to highlight. We have seen that in our entire universe, we have living things and life is present. Life is present in the universe. And that life comes in different forms, diverse forms, from the microscopic or unicellular that we discussed to what? Giant plants and animals. Then each organisms, and now organisms don't just exist, they exist in relationship with their internal body working and their external environment. The external environment. And then we'll begin to see how that will lead us to discussing some branches of life science like ecology. Like what? Ecology. And then we'll be talking about the ecosystem. The ecosystem. Eco from environment. So eco is the environment. So the ecosystem is a complex environment of living beings and their surroundings. And you have different sub-ecosystems in the universe. That's why they'll be talking about water animals. They'll be talking about land animals. They'll be talking about air animals, because these different environments are different. So this will be different ecosystems. And then even when we come to land, we begin to see desert. We begin to see grassland. We begin to see forest. And in forest, we begin to see rainforest. And we begin to see different types of forest. And then there can even be small things like river animals, like lake, like ponds, and like hills, plateau. You're going to see different smaller ecosystem, like your garden. So again, understanding life sciences will not only be about the life itself, it will be the animals, the plants, the micro, the macro, and their environment. So in studying life science, yes, we'll also meet the non-living aspect of it, because that will be the environment and the relationship with that. For example, when you look at plants, plants need what? Space. You cannot go and plant all your corn in one hole. You need space for them to grow. They need space to grow. Yes, they need space for them to feed in the soil, to take nutrients, water, to take air. They need space also above the ground, space in the ground. So again, they need water. Water is a non-living part. They need nutrients in the soil. They need air, which is also a non-living part. So again, we'll be looking at all this relationship together to form life science, even though the word is life. But in there, it is more complex than that. So let's take a dive in life science. In some subjects, they'll call it biology. But here we want to call it by its name, life science. So if we look at life science here, we'll see modern times. In the most recent biological developments, things that were thought to be impossible just 100 years ago become routine. Faulty body parts could be replaced by artificial replicas, and even genes could be changed to switch characteristics. There is something called genes. Genes. What are genes? What are genes? Genes are some special chemicals inside our nucleus of a cell that determines our behavior, determines our traits. Traits are your characteristics, shape, color, size, texture, all this, and also your behavior. So the way you look and how you behave, that is your traits. And the things that determines how unique that will be for every human is called the genes. And with genes are transferable from parents to offsprings. And parents also get it from their own parents. They also get it from their own parents' parents. So you see that it's transferred. So genes, we find genes in the nucleus of, and in the nucleus is a specialized part there called the chromosomes. And in the chromosomes, we have the DNA. So again, we are talking about this. So there's this unique thing that will begin to follow. And that today with science, we can change the genes. Oh my God, this is then the new world. American scientists discovered deep sea animals supported by the chemical energy of volcanic vents, the only life not dependent on the sun and photosynthesis. Whoa! I said all plants, all the food, everything you eat, the energy comes from the sun. And now they're saying that in the new world today, we've discovered that the heat inside the volcano vents can be used to sustain life. Volcano, we know volcano. Vents are like what? Vents are like openings. You know, little gaps where, the volcano lava can flow through like pipes in the rocks. Since it's not pipes, so those little paths, we call them what? Vents. Those little vents. So that's the vocabulary there. New life. Let's see again. The first human test tube baby. Woohoo! A baby in a test tube made with cells fertilized outside the human body. Woohoo! We say the sperm cell from the male and the egg cell from the female. The sperm cell will fertilize the egg, and then now they will exchange that chromosomes and become 91. That's why you get from your mom, you get from your dad. They say, but we can do all that was happening inside the ovum of a female. But they say now, hey, we can do it outside the human body. And that was that first test tube baby was born in 1978. Then in 1996, Dolly the sheep became the first mammal to be artificially cloned from body cells. From body cells, they make a full sheep, and they gave that sheep a name, Dolly. Cloning. Now, how do bacteria make each bacteria? The cells just split into two. You take the genetic material, the DNA material, and make a copy of that DNA material, and then it goes to different corners of the cell, and that genetic material form new nuclei. When the nucleus is plural, we call it nuclei. So from two nuclei, and then in the middle now, the cell membrane up here will dip, this one also dip, and then they'll split into two. So that is fission. That is what? Fission. Hey, separating. Coming together is fusion. Separating is fission. So now, in this fission, is that how cells multiply? Yeah, like blood cells. Your blood cells, if you say, hey, Falcon is growing, how do you grow? Your muscle cells make new muscle cells. How do they make new muscle cells? Like shitting babies like that? No. One just comes, and how do they split first? Before you split, you share your genetic material. Your genetic material goes because the heart of a cell is the nucleus. So two nuclei has to be made, and then now the cell membrane will contract and separate. Because animal cells, when we're talking about animal cells, animal cells don't have what? Cell walls. So we can only talk about cell membrane. So again, we can clone from cells. So if you take a cell, what cell? If you just take one cell, let's say the leg cell, can you make eyes, nose, and everything from the leg cell? No. So to make the sheep, how many cells did they take? All the cells from all the parts of the sheep? Yes. No. It's from one cell. So again, it's interesting to see how cloning was done. Well, that is science. So you see that in life science, there are exciting, unbelievable things that have been done, and then we begin to see them here. So that was 1996. Test tube baby was made, but that was like, okay, what happens inside the mother ovum, can it happen outside? So that was a good invention from science. And then now, can we make a sheep from a cell? That happened in 1996. Then let's see, artificial parts. Woohoo! Oh, we have artificial eye. Let's see, false limbs had been used since antiquity. In the ancient time, antiquity is a very good vocabulary. Antiquity means from ancient time. Antiquity. So false limbs. You see somebody walking like that, you say, hey, the person has two legs. If they open, you see bed, wood, or you see bed, steel, or you see bed, some other type of metal or plastic. So these are false limbs. Limbs are just examples of your extremities, like your hands and your feet. For animals that have four limbs and hind limbs. Four limbs are the front limbs, hind limbs are the back limbs. For a dog, you can't say hands and legs. You say forelimbs, F-O-R-E, and hind limbs, H-I-N-D. Forelimbs and hind limbs. Of course, you can use legs, forelegs and hind legs. So fore in front, like, and then hind behind. So you get that hind from behind, just that hind. So you know why we have the word behind, because of hind, and then forward because of fore, but then F-O-R-E, forelimbs, hind limbs. So we can make more in the 21st century. We have more sophisticated artificial body parts. And the first bionic, bio means what? Life. The first bionic eye is implanted in 2015. Interesting. So it's an eye that we make from machines, like this one. Okay. Now let's see. Fossils. More discoveries of ancient creatures, often preserved in amber, led scientists to new conclusions, such as the realization that many dinosaurs had feathers. Woohoo. So this is amber. Amber is a kind of place where things can be preserved without decomposition, right? So in amber, so that place where the thing can stay there without decomposition is called amber. Now, things decompose if there's heat, if there's air, if there's water. So those are the things that helps things to decompose. So for amber, for things not to decompose, you have to remove all the air there. You try to make sure that there's not heat. So you find a lot of things in icy region, things that have been preserved. That's why when you put things in the fridge, they will not decompose because it's cold. Heat will help speed chemical reaction. So if you remove heat, you cool down places, you slow down the process of things. All right, so we can really think in our head, how was the condition for things to decompose? If you plant your flower, if you plant your seeds, and if you want the seeds to germinate very fast, you put it in a cold place, no, it will not germinate. So if I put something in the soil, I take some plastic, I cover that soil. So I take some plastic, I cover the soil so that at night it will not heat up. So I cover it. Do you know why they put soil in those polythene bags? In those bags, they put soil in a bag and then plant inside so that it should be what? Warm and speed up what? Reaction inside. Will be heat. And then the seed will germinate. All right, so that's the kind of science that we have to put in our head. Practical science. Practical science. Very important. So there's something called the Human Genome Project. The Human Genome Project. A catalog of all human genes. It's called the Human Genome Project, HGP. Write it down. It's good to know these things. Some people just study science. These are some interesting things about science that you have to know. That should be like, motivating you to study more. The Human Genome Project is how to catalog all the human genes. If you see our genes, those compounds that make the genes, they give them some letters. So we have to catalog those letters to compare genes from one species to another. And because of that, they will know that, mosquitoes are not all the same. Mosquitoes are not all the same. They have the different genes. And then changing genes. In the late 20th century, what is the last year of the 20th century? 1999. Very good. So when you see 20th century, you have to go one number less before you start to count. 21st century starts from 20. 18th century starts from 17. 16th century starts from 15. So again, that's good. In the late 20th century, scientists became able to edit the genes of living things. What's the meaning of edit? To modify, to change, to enter or to change. Good. So scientists could enter something to introduce new genes and then change your genes. So living things. So that was in 2017. Some mosquitoes are genetically altered to try and stop the spread of disease malaria, the disease malaria. So how do they alter those mosquitoes? They alter them so that they cannot give birth. That's one way of altering them. So you have to go and find the gene that is responsible of birth. So if something cannot give birth again, we say that thing is sterile. Sterile. We use the vocabulary sterile. So there's a campaign called sterilization to make women not to give birth, called sterilization. Now, in the past, sterilization, so sterile. First, we look at the word sterile. So sterilization campaigns are campaigns that happen with some bad scientists in America. They were trying to get some African American or black women in the United States not to give birth. So they tried some experiments like this called sterilization experiments. In East Africa, in Kenya, there were also some sterilization experiments through vaccines. They give vaccines to girls, and when girls take those vaccines, the girls will become sterile. Sterilization. Sterilization. So again, science is supposed to help the world, but some mad scientists will use science to destroy the world. So again, we have to be very careful with this tool called science. So again, but for mosquitoes, so you see like the science is really improving. We can sterilize mosquitoes. We can make them not to give birth. But if you sterilize mosquito, you also change the ecosystem. Maybe these mosquitoes, or sometimes you make the mosquitoes to die young. You sterilize them, you make them to die young. Then if you sterilize them, it means they will not reproduce. They will not give birth to new mosquitoes. So what will happen? The mosquito population will reduce. Maybe you sterilize, but not all. The mosquito population will reduce. What if mosquitoes were responsible to eat some other harmful insects that also harm us? So if you sterilize the mosquito, you reduce the population, those harmful insects will also increase. Maybe, well, not about humans, maybe other things. Maybe these mosquitoes were helping to do some other things in the ecosystem to help bees, and bees comes and help plants so that we have food. So you see why by changing the population of mosquito, you can cause a chain reaction. What is chain reaction? Like a chain will affect one thing, that thing will also go and affect the other one. So we call that a chain reaction. Write it down, chain reaction, so that you can use that word, that phrase, chain reaction, when you discuss about things. So when there's a chain reaction, it means there's effect that goes like a primary. Before you get to the primary effect, you start from the tertiary, you go to the secondary, you get to the primary effect. So that becomes chain reaction. So here we've seen with our own eyes. We've seen science, life science, some really cool things about that. Now, any more cool things? Yes, we've seen animal behavior. Let's see, what about the structure of DNA? American and British scientists James Watson and Francis Crick identified that DNA, the genetic code of life packed into cell, has a double helix shape. What is a helix? Spiral, like a spiral. So it's a double spiral. So the other one is crossing like this, see? So this shape is called helix or spiral. So they discovered that the DNA has this shape. And you see now this shape, you see this color will be the same thing. This one and this green and this blue, they will be hooking like this. So they have names. They have names. This compound and this compound, they hook and meet. So adenosine, so let me get those four names of the DNA. So you have adenine, thymine, cytosine, and guanine. So let's get them here so that you can write them down. That DNA has this shape, and then the compounds that you see in the middle that are meeting to hold the helix together, there are four of them. Number one, adenine. Everybody say adenine. So we have adenine, and second one we call it thymine. Thymine. Adenine, thymine. Then we have cytosine. And the last one we have guanine. guanine. And yes, that is their letters. A, T, C, G. Yes. So they call them the base pairs. The base. But now we have the way that the pair adenine pairs with thymine, cytosine pairs with guanine. Adenine pairs with thymine. That's why you have them like this: adenine, thymine, cytosine, and guanine. So you see, like here, can you see the colors? That blue is always with green, red is always with yellow. Do you see any other way? There's no other way. So you see that I'm already teaching you this. So if you call this adenine, if this is A, then this one will be what? T, thymine. Then if this red is cytosine, then this yellow will be what? Guanine. So that's how you put these pairs. You put your A is pairing with T. And to put a pair, so it's just like in your chemistry, but did you know that these are still compounds? So this is still chemistry that we are seeing here. These names are compounds. We can find their elements. This is just their common names. So adenine and thymine, cytosine and guanine. So that's how they pair to hold the double helix together. And this double helix is also a chemical compound. So this helix will be like carbon, hydrogen. These are bonds. These are bonds. So it's a giant molecule. DNA is a whole giant molecule, very long, made up of what? Hundreds and hundreds of atoms connected together. Then this adenine and thymine, they are also chemical compounds too, holding in the middle. And they say this is where the meaning of life is, in DNA. That life is in DNA. The code, genetic code of life is packed into cell. And what they call it is called information. Like if something can determine your behavior, your character, your traits, how you look like. So it means there's information stored in these chemical compounds. So very unique. So that's why the structure of the DNA was very great, a discovery to determine the structure of the DNA, to find this double helix. So the bonds that hold adenine and thymine together is 2 hydrogen bonds. The bonds that hold cytosine and guanine together, 3 hydrogen bonds. So you see, you study chemistry, and that is the chemistry. So what else? Every matter is made up of all these elements. So whatever we are talking about here is what? It still goes back to your chemistry. All right. So again, we should enjoy this as something that we study with pleasure. Now, the rise of ecology. The rise of ecology. The study of ecology. What is ecology? How organisms interact with their surroundings. Emerges in the 1930s. In the 1930s. The 1930s are which year to which year? Very good. From 1930 to 1939, to the end of the 1939. So those are the years of the 1930s. And why do they say, why not say in the 30s? Because every century has its own 30s. So here you have to specify which century. 1930s is in which century? In the 20th century. Excellent. So as British botanist, Arthur Tansley introduces the idea of ecosystems. Ecosystem. When you break the ecosystem into a system, is something built up of many parts. organisms, different organisms in different environments. That's how we build. In those environments, you have water, rocks, earth, soil. We have those things that we studied yesterday also. So that is that, the rise of ecology. So ecology is one way to look at life. Antibiotics. What are antibiotics? Very good. So antibiotics were discovered. British biologist Alexander Fleming discovers that a substance, penicillin, the first known antibiotic, stops the growth of microbes. Antibiotics. Anti, bio means what? Life. Anti-life. It stops life. From there you can find the meaning of the word. Anti-bio. Antibiotics. So, are now used to treat many bacterial infections. If you have a wound, oh yes, take your penicillin, and rub it there, or you drink it. It will go now into your cell and goes to those areas where there are foreign organisms, and it will stop them from multiplying because its name is what? An antibiotics. All right. We go next. Chromosomes. Chromosomes and genes. How do you understand them? Genes live in chromosomes. Chromosomes themselves can be thought of as genes. American scientist Thomas Hunt Morgan carries out experiments on fruit flies, which prove that the unit of inheritance are carried as genes on chromosomes. So very good. So chromosomes became a very important part, and chromosomes, they live in the nuclei of cells. Early 20th century, what happened? Better microscopes and advances in studying the chemical makeup of cells help to show how all life carries a set of building instructions in the form of chromosomes and DNA, while ecology and behavior became new topics of focus. All that happened in the early 20th century, meaning that in the 1900s, or the 1910s, or the 1920s, or the 1930s. Those are the early 20th century years. Oh yes, microbes. What are microbes? An experiment by the French biologist Louis Pasteur. We have the Pasteur Center right there at the central town. Proves microbes are sources of infection. It also disproves a popular theory which had argued that living organisms could be spontaneously generated from non-living matter. Hey, so you just take some non-living matter and then you put them together, and then you have some living things. So before, that's what people were thinking. So he did some experiment to show that infections that come from microbes. People get sick because some microorganisms are introduced in your body. Those are the things that get you to fall sick. But some people will just think like people just fall sick for other reasons. But this guy discovered that you fall sick because of microbes. Like what? Bacteria, viruses, what? Plasmodium. Like what causes malaria is called plasmodium. So you have other microbes, other names of microbes. All right. So let's see. Evolution. The father of evolution is called Charles Darwin. British biologist Charles Darwin published a book on the origin of species. Explaining how life on Earth has evolved by natural selection. What is natural selection? That we, over time, the survivor of the fittest. Now, if in this place is a harsh environment, if we take all of us, they put us in a desert, and then in that desert, only the fittest person, the strongest person, the person that can adapt better in that environment will survive. So the one that is surviving means is the one that has been able to be selected by that natural environment to survive. 5, and we call that natural selection. Because of the condition in the environment, some organisms will die and some will survive. So that is called natural selection. So that is how we evolve. And that is really true in many ways. Because in the beginning of creation of life, there weren't humans. Humans came later. Humans came much more later. And long ago, 65 million years ago, we had dinosaurs. Dinosaurs are no more. So the natural environment, there are things that happen in the natural environment to select. And every year, species are going out of existence. And we call that extinction. Extinction. To go out of existence. Entire species will go out of existence. We call that extinction. So we have extinction events every year. So you can read more about extinction. And then we say, hey, some species are endangered. Endangered species. Endangered species like panda. We have very few hundreds of panda alive left on the whole planet. Panda. Kangaroos, young man? Yes, kangaroos too. Kangaroos because they live in Australia, but we breed them. So humans have to intervene to make more kangaroos. Like panda also, they have a park where they groom the panda to make new baby pandas. And for pandas to, even for the male to come with the female to give birth, they don't love each other. They don't like each other at all. So the scientists there, they have to do so many gymnastics. You can also watch videos on YouTube how pandas are bred. So endangered, from this danger, we say endangered, face danger of extinction. So we call those endangered species. Well, and then we have some, with this ecosystem environment, we have some forests, like the tropical forest, the Amazon rainforest. The forest in Cameroon, there's certain places in Cameroon where people cannot go there and bring down the trees. Like we call those parks, the Koro Park and the Waza Park. So we preserve those areas so that all the organisms living in that place will not lose their natural environment. And we have other things that are happening to make the ecosystem, oh my God, to get the situation to get worse and worse. For example, like forestation, like deforestation and desertification. Desertification means that the desert is expanding. We call that desertification of the soil. So when our natural soil is made up of what? Dead plants and animals. But when in the desert there, you don't have any dead plants. The desert there is just made up of what? Sandy soil, right? So the desert is eating and coming to land. And then what increases desertification also is the cutting down of trees. When you cut down trees, the sun hits the ground, and with that heat, all these decomposed things will all decompose, and they go into the nitrogen cycle or the carbon cycle. And you have learned all those carbon cycle, nitrogen cycle. You have your cycles, nitrogen cycles and carbon cycles. So you look at those things, you will see how nitrogen from the air, how the cycle nitrogen goes from fertilizers and all those things. You see the movement of nitrogen in nature. So those things are good to learn. Water cycle, carbon cycle, nitrogen cycle. So we've seen all this. Inheritance. Let's look at inheritance. What's the meaning of to inherit? To take over. Very good. Or for a gene to move down the family line. So the children will take over the gene from their parents. So that's why we say people live forever. Even though your daddy will die one day, but the daddy's gene did not die. But the gene was not the daddy's. It was the daddy's, mommy's. So that's how people live forever. But because the gene never dies, the gene moves from. They say, okay, what if the last person did not give birth? Oh, yeah, the last person did not give birth and dies, so it is gone. Are you sure? What if the first person made 2 sons, and these 2 sons they made many, many sons? So in that tree, how many people are there? How many children are there in the tree? And if that tree is going, it's multiplying exponentially. So you started with one ancestor, gave birth to 2. Then 3, 4. 3 gave birth to what? 10. 10 gave birth to what? 300. 300 gave birth to what? So how can you kill a gene? Oh no, Sacramento. So an Australian-born monk called Gregor Mendel carries out breeding experiments with pea plants that helps to explain the inheritance of characteristics. So he takes a pea plant. We have different types of pea, maybe the red pea and the green pea, and then he will use the pollen dust from the red pea to pollinate the white pea, and then they will make new pea plants, and then they will make new seeds, right? And then plant the seeds, and then the pea that will come out of that seed will be what? Will be red and white together. Yeah. You take a black woman and a white man, they have a baby is what? Mixed child. Is the color totally black? Is the color totally white? Then what are you talking about? You take a short man and a tall man and they have a baby, and the baby is where? In the middle. But if the tall man's gene is stronger, the baby will be what? Tall. So that is what Mendel did. Mendel took genes. And then that's when you see equation. How they'll say, you want to see genes?
Yes.
Oh, yes. So in genes, you can say, okay, 1 pea plant, the gene was big purple pea and small purple. And then the other one was big yellow and. So how do they come together? This one, big P, and this one, big P also and small p. Small p blue and then you come and draw it. That is how you do gene equation. Oh. You have seen that somewhere? Somebody say, oh yeah. Hey, Lorosokoto. Now you understand the genes? Oh yes, XY, those are genes. So this one comes with a yellow, big P, small p. Which one Aquila wants to be? Small p, small p?
Big P.
So the big P, we call it dominant gene. Everybody say dominant gene.
Dominant gene.
And the small p is called recessive gene.
Recessive gene.
Very good. So black people have dominant gene. So that is why we have melanin stronger. Oh yes, black people we have dominant. White people have recessive. Our genes are stronger than them. If you have a white baby and a black baby, the same day born, the same day, the black baby will be the first to crawl, will be the first to stand, will be the first to walk, will be the first to jump. Oh yes, because of the dominant gene. So if you want to know why black people are the most hated people in the world, it's because we are naturally the best. Oh yes. You go to football, the best players in the world, they'll be black. You go to running, the fastest person in the world, is black. You go to brains, the smartest person in the world who made a supercomputer, 2 Nigerians, black. You go to anywhere, you meet black. You want to find the most stupid person, will be a black. The most intelligent person will be a black. So black people, they form all the spectrum from the smartest to the stupidest. Oh yes. All right. So don't say that, hey, I'm black, then I'll be the best. If you don't study, you'll be the stupidest person ever. So you have to watch out. All right. So what else can we see in life, discovering life? Okay, discovering life. Now we have seen genes, so we know more about that. We've seen our DNA. We have seen the base pairs that holds the double helix together. We have seen adenine, thymine, cytosine, and guanine. Discovering life. Ever since people first began to observe the natural world around them, they have been making discoveries about life and living things. Biology, the scientific study of life, emerged in the ancient world when philosophers studied the diversity in life's creatures, and medical experts of the day dissected bodies to see how they work. So we can also take a frog and we operate it and open it. That is called dissection. Hundreds of years later, the invention of the microscope opened up the world of cells and microbes and allowed scientists to understand the workings of life at the most basic level, at the cellular level. At the same time, new insights helped biologists answer some of the biggest questions of all: the cause of disease and how life reproduces. How to make new cells, how to make new bones, how to make new kinds. Oh, yes. So that is that. For that, we have seen that timeline of discoveries, antiquity to the 16th century, describing fossils. Fossils are remains of what? Dead plants and animals, or architecture. So it's not only dead plants and animals. Maybe the way people built structures in the past, maybe we can find that pictures like the remain of that. So fossil is not only the remains of ancient plants and animals, it can also be about architecture or some tools, like the weapons that early men used. We can find those spears, the remain of that spear preserved inside ice or glaciers, or in the rock, sedimentary rocks. So we have to complete our definition of fossils. Not only living things, but you can also have tools.
So how are machines going to know how many years?
Well, it's called carbon dating. All again from your chemistry. That when we see a plant, we can see the concentration of carbon atoms in it, a special isotope called carbon-14. If you look at a plant, you can know, like in a healthy plant, how many carbon-14 are inside the carbon. There's carbon-12 and carbon-14, so it's called the isotope of carbon. Then if you look at a plant, a young plant, we can see the number of carbon-14. You look at a plant of 60 years of age, the carbon-14 will reduce. You look at a plant of 200 years, the carbon-14 will reduce. So from there, we just study now how much the carbon-14 reduces with time. So when you find a fossil, you just pick it, take it to the lab, you check the amount of carbon-14 inside, and then you can date how many years back. It's called carbon dating. Carbon dating. Oh yes, science. Everything science, not magic. Oh yes. Healing theories. Early anatomy of the human body, cataloging life, different life, classifying different species. Vaccines were invented, photosynthesis, we study more of that. Classifying life, looking at microscopic life, we look at that already. Blood circulation in the heart, the arteries and the veins. The ones that are carrying blue blood are the veins. The one carrying red blood are the arteries. So again, and 4 chambers. And blue blood means dirty blood, young man, dirty, and carrying carbon dioxide, rich in carbon dioxide. And the red blood rich in oxygen. All right, taking it to every cell. Then the blue taking, it's just the color that they put there, but then it just means dirty blood. So it's just color code for you to understand. All right. And then you see how each of them, they go into the cells. The blue and the red, they go to the cells. The red is supplying fresh blood rich in oxygen. And then the cell will the cell will use up that oxygen and send out carbon dioxide. And then the other one will carry it. So the carbon dioxide one is the one that they color blue. So here it's not that it's blue. It's not that it is really blue, even though some animals have blue blood. So this first page, we have seen some summary of life science. Exciting things and some not too exciting things. Some discoveries, some scientists' names, like Louis Pasteur, like Mendel for hereditary for gene experiments. Mendel trying to do recessive and dominant gene to put different genes together and like that, and you study more of that. You've seen Darwin for evolution. Darwin, we've seen Darwin is the name of a scientist. We've seen Louis Pasteur, who studied diseases and created penicillin, which is an antibiotic. Yes, so we've seen all that. So before now, we enter now into what is life. All right, for today, we can end here. Tomorrow is another day. All right. Let's rumble and vamonos. Did anybody join this class? Any other student in this class? No?
Let us stop the video. Goodbye.
Cassava.
Besides,